Great. Hello, everyone. Thanks for taking the time to come to my presentation today. Today, I'm going to be talking about how we've been working to quantify absolute metabolite concentrations in T cells using a novel method that we're terming shotgun metabolomics. So there are approximately 200 distinct cell types in the human body. All of these cells have different functions, phenotypes, and metabolic needs. In particular, we're interested in blood cells because they're the only cells that we can remove from a patient's body minimally invasively, then we can engineer them and reimplant them. And so we need a comprehensive and quantitative method to really quickly characterize their metabolism. And my project in particular has been focused on T cells. T cells are antigen-specific immune cells, which allow an immune response to virus-infected cells. They originate from stem cells in the bone marrow. They migrate to the thymus, where they mature and proliferate into different parts of our body. Initially, these are naive cells, but after activation, they proliferate into memory and effector T cells that can only respond to specific antigens. And there's a few different types of T cells. There are helper T cells, which recognize infected cells and organize an immune response. Then there are killer T cells, which recognize an antigen and release cytotoxins to kill the infected cells. And then there are also regulatory T cells, which modulate the immune system. T cells rely on a highly dynamic metabolism and metabolic cues to provide the bioenergetic requirements for rapid proliferation and immune response. This is really powered by metabolism. So on the metabolic scale, this broadly translates to a shift from heavy reliance on oxidative phosphorylation pathway at quiescent state for all of their ATP and energy needs, to a shift towards aerobic glycolysis when they need to proliferate, which, while that produces less ATP, gives them more of the intermediates that they need for rapid proliferation. And then when they no longer need to proliferate, they shift back into this kind of more dormant state, and they go back to memory cells, where they rely primarily on fatty acid oxidation and oxidative phosphorylation for all of their energetic needs. Metabolism has been identified as an important component of both immunosuppressants and successful cancer therapies, which is why we want to be able to characterize T cell metabolism. Thus far, a lot of the research on T cell metabolism has relied on relative metabolite levels, which, while those paint a picture of the broad metabolic shifts in T cells, a more quantitative understanding of T cell metabolism is needed for thermodynamic, kinetic, and flux calculations. So just to illustrate that, I have here the how you would write out the equation for a reaction rate or a flux. And this flux is dependent on the enzyme concentration, as well as the probability of substrate binding, and then also thermodynamic reversibility. And what you'll notice in this equation is a lot of absolute concentration. So if we have absolute concentrations, we can plug them into this equation and build more predictive models of T cell kinetics. That's all great, but traditional absolute metabolite quantitation is very labor intensive and expensive. So traditionally, how our lab and other labs would do this is they would buy a chemical standard of a metabolite they're interested in, for example, ATP. They would add a known concentration of that into the extraction solvent. We use acetonitrile, methanol, and water. And then they would grow the cells that they're interested in fully labeled 13C carbon media. So they would either use U13C glucose, or if they're growing mammalian cells, also U13C glutamine. And they would extract these cells into this extraction solvent containing the chemical standard. And what they end up with is a sample containing a mixture of intracellular metabolites, which they know are going to be 13C labeled, and the standard, which they know will be unlabeled. And so with LCMS, we're able to distinguish the two peaks. And since we know the concentration of the standard, then we're able to extrapolate the concentration in of the metabolite in the cells. This works very well. However, it can be very expensive. If you're interested in a wide range of metabolites, it can also be extremely time consuming. Fortunately, we have data for about 150 metabolite concentrations in 3 model organisms, E. coli, yeast, and epithelial cells. This was very time consuming data to collect that Dr. Park and his colleagues worked on. But our plan is to use that data and use those cell types that we already have data in as the internal standard instead of fine chemical standards. So our new approach uses the reference cell type as the standard. So we'll grow the cells that we're interested in this case, T cells in unlabeled media, and then we'll grow whichever reference cell type, E. coli or IBMK, baby mouse kidney cells, in a fully labeled media. And then we're able to extrapolate these same ratios like we did before, but for a really wide range of metabolites. And we know all the concentrations that come from the 13C labeling because those are the ones that come from the cells that we already have data on. And then from there, we're able to extrapolate the concentrations in T cells. So to give you an idea of what this would look like, here is ATP. ATP has 10 carbons, so if it's fully unlabeled, that would read out as N+0 on the LCMS. If all of the carbons are fully labeled, that would be N+10. Because of impurities in the isotope and then also natural 13C abundance, we tend to get some smaller peaks at N+1 and then N+8 and N+9. So we group these together and we have an unlabeled fraction and a labeled fraction, and we know that the unlabeled fraction corresponds to the metabolites in the T cells, and the labeled fraction corresponds to the metabolites in the reference cells, which we already know. So this co-extraction technique enables metabolite quantitation en masse, so we get a lot of different metabolites at once rather than just one. And so the way that we've been doing this is we vacuum filter. We put a filter on top of an Erlenmeyer flask, which is attached to a vacuum, and we're able to filter out droplets of E. coli culture grown in fully labeled media and T cells, and then we extract them simultaneously and end up with a mixture in our sample. We have also developed this to use with a cell type that's grown adherently. So baby mouse kidney cells, which we have data for, are grown adherently on a plate, and so we combine the suspension cell extraction technique that we have with the adherent one, and similarly get a mixture coming from each cell type. So before we started to apply this with T cells, we wanted to verify that the method would actually work. And so the first thing that we did is that we grew 2 different cultures of E. coli, one in fully unlabeled media and one in fully labeled media. And we extracted them at known ratios and verified that the metabolite ratio, the ion counts that we read out from the LCMS for a bunch of different metabolites matched closely to the ratios that we expect. And sure enough, that's what we saw. So these lines represent the ratios that the cultures were extracted at, and then the dots represent different LCMS reads from different metabolites and showing which ion counts they have from labeled and which ones they have from unlabeled. And as you can see, there's a lot of agreement across most of the data. We did see higher deviation at this 20-to-1 ratio in green. And so we decided to only consider data points that fall within this 1-to-20 ratio here. We similarly wanted to validate the combined suspension and adherent method. And so we use baby mouse kidney cells as the standard to remeasure concentrations in E. coli. And this data wasn't as good. So we still see it's still around the line of unity, but there's a lot more deviation, particularly in the metabolites where the concentrations from 2016 had higher errors. So with this in mind, we decided to only use IBMK-D3 as a standard for concentrations where we don't have existing data in E. coli and there's no other way to do it. But other than that, we try to rely solely on E. coli. So once we validated that, we were ready to move on to working with T cells. We worked with Jurkat cells, which is an immortalized helper T cell line. And we were a little bit worried that the extraction conditions could greatly affect the quality of our measurements, particularly for T cells, which are not as robust as E. coli. And one thing that we're really worried about was that the vacuum pressure might lyse the cells before extraction. So we tested a variety of different vacuum pressures. And what we actually found was that for amino acids, which are found in media, higher vacuum pressure led to lower peak levels, which actually indicated more media absorption, which is a good thing. And then for intracellular metabolites, we saw just more consistent measurements across the board at higher pressures, probably due to the fact that the extraction occurred more rapidly because it was higher. So we actually decided to work within this -14 to -20 inches mercury pressure range. But what we did realize was that typically we like to rinse the cells because, like I mentioned, the media has amino acids on it, and not all of it gets filtered through the filter. And so normally labs will either rinse with ammonium acetate or PBS. But what we found was that rinsing with ammonium acetate or PBS greatly affected the peak quality for T cells. And so we realized that we wouldn't be able to do that. And so now in our method, we account for the amino acids by doing control samples where we just filter the media and kind of back calculate. With all of that, we were able to measure 79 metabolites in the Jurkat cell lines, and we compared that to existing data for the 2 model organisms that we used as reference. What we saw was a lot of similarity across the 3 organisms, particularly in the grouping. So like the highest concentration group of metabolites across all 3 is amino acids. We also plotted the different metabolite concentrations between different organisms and found pretty high correlation across them. But then we also see some kind of distinct features across different metabolites, particularly this higher valine concentration in T cells, which might be interesting. As I mentioned before, we want to actually use these metabolite concentrations to get a sense of like function and kinetics. And so one thing that we could do is we plotted the concentration of different metabolites across their Km values for different reactions and also their Ki values. And what we saw was that 63% of the metabolite concentrations lied above the Km values for metabolic reactions, which indicated a high enzyme substrate binding site occupancy and efficient enzyme usage. Conversely, less than 50% of reactions that had inhibitor concentrations above the Ki values, which indicated that inhibitor sites are less saturated than enzyme binding sites. So this is just to give you an idea of how someone could use these concentration measurements to get really predictive information. Now that we've worked with Jurkat cells, we would like to also quantify metabolite concentrations in primary CD4 and CD8 T cells. And we have a few key questions that we'd like to address. The first is whether or not Jurkat cells are a reliable metabolic model for T cells. Like I mentioned, they're an immortalized cell line, so they're really great for culturing in vitro, but we're not sure how well they represent primary T cell metabolism. We also want to know how much variability there is across different donors. And we're also interested in seeing how much variability there is between different T cell types, particularly CD4 and CD8 T cells, which are helper and killer T cells, respectively. So this week, I got to do this for the first time with with one donor. So I got PBMCs and I was able to isolate CD4 plus and CD8 plus populations and do shotgun metabolomics with them. So I've got some preliminary data. The first interesting piece of data that I found was with the 30 or so metabolite concentrations that I've measured thus far. I found really strong correlation across both the CD4 plus and the CD8 plus metabolite concentrations. This is a lot higher than the correlations that we saw across different organisms. So this is very interesting. It might suggest that there's very little difference between CD4 and CD8 T cell metabolism, and perhaps some of these outliers might be interesting to look at. I also plotted the CD4 against the Jurkat cell concentrations, and we saw significantly worse correlation here at 0.70. And so this, at least the first donor, suggests that Jurkat cells are probably not a very good or reliable model for T cell metabolism. As I talked about today, quantitative metabolomics in primary cells requires accessible and high throughput techniques. Shotgun metabolomics is a robust approach for quantifying absolute metabolite concentrations. These can be used to infer kinetic information. The preliminary results that I have from the first donor suggest strong correlation between CD4 and CD8 cells, but it also reveals some limitations in the Jurkat cell model that we're going to explore. I've presented today on T cells, but really we want shotgun metabolomics to be easily adapted to any cell type of interest so that these quantitative metabolomics measurements are more accessible to a lot of different fields and different labs. So I'd like to thank my lab for all of their help on this, and also QCBio for having me today, and I'd be happy to take any questions.